Hello, my dear students. Today we are going to discuss about the neurotransmitters mechanism of action. Here, the neurotransmitters can act both type. Either they can type the directly, and either they can act indirectly. So here, the mechanism of action in this lecture we will discuss about the mechanism of action. The neurotransmitters mechanism of action is that in the short form I will write the neurotransmitters is that NT. The neurotransmitters can act directly, and second form in the either they can act directly or they can act the indirectly. Okay, so here in the direct mechanism of action or the direct action, the directly is also termed as direct action. In the direct mechanism of action, how they will act, the neurotransmitters will bind. The neurotransmitters bind to an inotropic receptors, bind to an inotropic receptors, inotropic receptor. After binding, what will happen? After binding, that they act, means after binding, they act quickly to open or close the ion channels. Means when the neurotransmitters bind to an inotropic receptor, they either open or close the particular ion channels. Open or close particular ion channels in the membrane. Okay, ion channels in the membrane. Okay, so as the particular ion channels open or close, then what will happen? This will affect the membrane potential. This will affect the membrane potential. This will affect the membrane potential. Okay. And as the membrane potential will affect or will change, what happen? Either the excitation or inhibition. Both can means at a time, either the inhibition or excitation will take place. Okay, inhibition. Like that. Okay, so in the neurotransmitters, I will just draw the diagram. Suppose that this is the membrane and this is the inotropic receptors, and as the neurotransmitter will bind, what happen? These neurotransmitters will, these inotropic channels open like that. Okay, as they open, particular flow of ions, they allow either the particular ions or they can stop the particular ion. Okay, like that they can act. Like that they can change the membrane potential, and according to the membrane potential, excitation or the inhibition take place. Okay, understood? Now another thing is which type of the neurotransmitter will act like this. The example is that acetylcholine, and some amino acids, like that. Okay, now come to the indirect mechanism of action. Here I will attach the new slide. Okay, now come to the indirect action. Here, come to the indirect action. As the name suggests, what will happen? As the name suggests, the mechanism of the action or the neurotransmitters will generate the mechanism of action indirectly. means they will not proceed directly via receptors. They will generate or they will activate the some another molecules. They are named as the second messenger, like G protein, like cyclic GMP. Such proteins or the such molecules will activate, and after activating these second messengers, what will happen? They influence the chemical reaction inside the cells, and such chemical reaction will proceed the inhibition or the excitation. Understood? So in the indirect action, what will happen? The neurotransmitter will bind with receptor, okay? And after binding, they activate. After binding, they activate second messengers like G protein, like cyclic GMP, like that. Okay. And after activating the second messengers, these second messenger molecules influence the chemical reaction. These second messenger molecules influence the chemical reactions. Chemical reactions inside the cells. And as this such chemical reactions proceed, what will happen? These chemical reaction either can produce the excitation or the inhibition of the post-synaptic neuron. Post-synaptic neuron. Okay, getting my point. So here, the indirect action, the neurotransmitters bind with the receptors, and after binding the receptors, they activate the second messenger molecules like the G protein and the cyclic GMP. And these second messengers influence the particular chemical reactions inside the cells. And after these chemical reactions, the excitation or the inhibition of the postsynaptic neurons occur. So like that, they can act the indirectly. So many neurotransmitters act like that. For example, that is monoamines, neuropeptides, like that. Carbon monoxide also behaving like indirect action. Okay, so this is the complete mechanism of action of the neurotransmitters. Now we will discuss in the same lecture continuously. We will discuss about the removal of neurotransmitter. The removal means the removal means the neurotransmitters when released in the synaptic cleft, as the membrane potential will change, or as the impulse propagate further, then what will happen about the neurotransmitters? First of all, these neurotransmitters remove with the three mechanisms. First, either they diffuse out, and second, they're degraded by a different type of the enzymes, or they can uptake by the different transporter proteins back to the presynaptic neuron. So here in this lecture, we will in this part of the lecture, we will discuss about the removal of neurotransmitters. Removal of neurotransmitters. Okay, here the neurotransmitter removal, why is it essential? As the impulse will propagate further, if the neurotransmitter will remain there, what will happen? The message continuously proceed. Already the message will go on, proceed in the postsynaptic side. But as the neurotransmitter remain in the synaptic cleft, the again and again will generate the new and new messages. And what will happen? This will proceed the impulses and the series of impulses, and this will cause a diseased condition. So after after propagating the impulse or after propagating the action potential, the neurotransmitters have to remove from the particular site or from the synaptic cleft. So neurotransmitter removal can be removed by the three ways. First is by the diffusion. Some released neurotransmitters can diffuse away from the synaptic cleft. Some released neurotransmitters NT in the short form, neurotransmitter molecules can diffuse away from the synaptic cleft. Means easily they can swipe or they can diffuse away from the synaptic cleft, so that they cannot remain in the synaptic cleft and they will not generate the impulses again and again. Okay, away from the synaptic cleft. Like that. Now the second mechanism is that enzymatic degradation. Enzymatic degradation. What it means, as the action potential will generate or will proceed towards the postsynaptic neuron, the neurotransmitter will degrade or will inactivate through the different enzyme. Like that, in the acetylcholine, means as the acetylcholine generate the action potential or the impulse towards the postsynaptic site, what happened of the acetylcholine in the synaptic cleft? The acetylcholine will degrade into the acetate and the choline by the enzyme acetylcholine esterase. Okay, so acetylcholine will degrade, so it will not activate or it will not produce any kind of the action potential. Okay, so here the acetylcholine, in the short form, the acetylcholine represent as ACH. Acetylcholine degraded by acetylcholine esterase. Acetylcholine esterase. Okay, and this acetylcholine esterase will degrade choline and acetate molecules. Getting my point? This is the enzymatic degradation. Now come to the third removal way, that is the uptake by the cells. Uptake by the cells, here, uptake by the cells or for neurotransmitters will take by particular transporter proteins to the presynaptic site. Okay, so this is the third removal way, that is the uptake by cells. Uptake by cells. Okay, here many neurotransmitters are actively uptake back into the neuron that released the membrane protein do this job, they do this uptaking job. Such proteins which are act like that, they are known as the neurotransmitter transporter proteins or the neurotransmitter transporters. Okay, so some neurotransmitters here, some neurotransmitters are actively taken up by or the uptake back into neuron via transporter proteins. Okay. What kind of the neurotransmitters or the example of the neurotransmitter which are actively taken up by the transporter proteins? For example, that is the norepinephrine. The example, that is the norepinephrine. Okay, here the norepinephrine's are the neurotransmitter which are actively taken up back into the neurons by the transporter proteins. This characteristic is the transporter proteins mechanism is using to remain means we are using some drugs. What I'm going to tell, it's a kind of the just knowledge purpose. Some drugs are blocking such uptaker proteins, or we can say transporter proteins. And when such drugs block these transporter proteins, they enhance or prolong the activity of the neurotransmitters. Okay, so just like one drug, that is the fluoxetine or Prozac. The brand name is a Prozac, and this is the selective serotonin reuptake inhibitor. When these drugs are used, this drug will inhibit the serotonin transporters, and as these drugs block the serotonin transporters, the serotonin activity will prolong at the synapse. Okay, so if some drugs, if we are blocking some transporter proteins, by blocking these transporter proteins, the neurotransmitters activity prolong, and this neurotransmitter will produce a prolonged action or an action for a long time. These drugs are used for, which are I have named as the fluoxetine, Prozac. These drugs are used for the like bipolar depression, like the obsessive compulsive disorder, like the depression. So just for knowledge purpose, I have given here that some drugs inhibit these transporter proteins. Okay, what will happen? For example, I'm writing the name. That drug is the fluoxetine. The brand name, that is the Prozac. This drug will inhibit the serotonin, inhibit serotonin transporters. Okay, means fluoxetine is the selective serotonin reuptake inhibitor drugs. It is the selective serotonin reuptake inhibitor drugs. Inhibitors. Okay, so when the fluoxetine is used, it inhibits the transporter proteins, and as serotonin transport, as they block the serotonin transporter proteins, they prolong the synapse serotonin activity, prolong the serotonin activity at the synapse. This serotonin activity as the increase, so the fluoxetine drugs is used for the treatment of depression, or the obsessive compulsive disorder, that is the OCD, bipolar depression, and many more things. Okay, like that. So in this lecture, we have studied about the mechanism of action. I will summarize. The mechanism of actions, the neurotransmitters can act directly or indirectly. Directly, they can act on the inotropic receptors, and they open or close the particular channels. Indirectly, they can act by the second messengers. The removal of neurotransmitters can proceed by three mechanisms. Either the first, they can diffuse out, or second, they can deactivate it by the particular enzyme, like the acetylcholine esterase deactivate the acetylcholine into the acetate and the choline. And third one, that is the uptaker proteins. Such uptaker proteins uptake the neurotransmitters from the synaptic cleft and just stop these neurotransmitters action. Some drugs We can use drugs for blocking such transporter proteins for prolong the action or prolong the mechanism of action of particular neurotransmitters. Such synaptic, for example, that is a fluoxetine or the Prozac. These drugs inhibit the serotonin transporters, so it prolongs the serotonin activity at the synapse. And these drugs used in the depression, obsessive compulsive disorder, bipolar depression. I hope you find this lecture informative. If you find so, kindly just share this lecture. Till then, bye-bye. Thank you.